Welcome to my presentation today. In this short talk, I will seek to highlight some of the aspects of extractables and leachables which have changed over the last 30 years and some of the things that have remained the same. In describing the last 30 years of activity in extractables and leachables for inhalation products, clearly it's going to be a summary and a rapid journey through topics that continue to develop and have certainly maintained my interest throughout the time I've worked in this area. I would like to start this journey with a substance group that perhaps kicked off regulatory interest in leachables from inhalation products, and certainly was my introduction to this area in the 1990s. As a 20-something analytical chemist, I was first introduced to this topic via my involvement in the development of methods for polynuclear aromatic hydrocarbons, or PAHs. These compounds are so called due to their structural feature of multiple fused aromatic rings.
I was involved in two styles of analysis for PAHs at the time, an HPLC method using a UV detector, and a newly introduced gas chromatography method with a shiny new benchtop mass spectrometer. The model I used I saw recently on eBay for £858, but at the time, no doubt it was a significant investment for the department. This presentation will consider E&L in all inhalation products, but I will highlight that Metadose inhalers have always presented a very particular and somewhat unique risk from leachables. This risk has a number of interwoven factors which account for this. Number 1. The route of administration. Dosing directly to the lungs, these being a potentially diseased organ. Number 2. The typical disease type or dosing regime, potentially requiring continuous daily or more dosing over many years. And number 3. A complex container closure system or device consisting of several materials, a can made of metal, a valve, elastomers and plastics, an actuator with some other plastics, and an overwrap that may be made from laminate foils. And lastly, a set of excipients which are highly organic in nature, including a volatile organic propellant, and thus a good extraction solvent for organics within the molecules found in the plastics and elastomers, much more than water would be. Thus, this combination of factors places the MDI in a unique risk category for leachables, and during the period from the mid-90s when the FDA was writing its packaging guidance, this was certainly a major factor in their thinking and perhaps resulted in an overestimation of the risks from other inhalation products such as an aqueous nasal spray or a dry powder inhaler. The original risk matrix in the 1999 packaging guidance stood for many years as the last word on leachable risk. It was modified and represented in the USP's 1664 publication in 2015, and you will see that the risks from dry powder inhalation products and aqueous nasal sprays were reappraised and reduced to more correctly position their relative risk. At the beginning of the 21st century, the National Toxicological Program published a vision for toxicology in the 21st century with the goal to develop a more efficient and effective approach to predicting how substances impact human health. This was called Tox 21. Returning to the 2006 PQRI document, a great leap forward was made with the introduction of a risk-based safety concern threshold, which for the first time provided for a reasoned limit to the leachable safety assessment in inhalation products on the basis of a toxicological risk and not on the basis of analytical capability. In July 2020, the USP produced a stimuli article entitled USP Approach to Future Revisions of Biological Reactivity Chapters 87, 88 and 1031. It should be noted that the current USP chapter on biological reactivity is very old, the in vitro chapter 87 being unchanged since it was added in 1990, and the in vivo chapter predates the numbering system which began in 1975. As we move forward, we now need to have a clear science-led toxicological approach which can provide information and assurances that leachables are not a safety risk, and the development of a valid toxicological approach is certainly needed. The reasons for ICH-Q3E are clear to me. No internationally harmonized guidance on E&L assessment and control exists. Specifically, no international guidance that addresses the multiple therapeutic modalities and dose forms, including identification, qualification and reporting thresholds, and also addresses safety assessment. The alignments against the principles of a safety-based, risk-based and quality by design approaches are not currently available. These current gaps generate uncertainty for industry and regulators due to the lack of clarity regarding extractables and leachables to meet the regulatory expectation. The uncertainty promotes potential delays in the approval of regulatory applications and can lead to variable interpretation. As I have been illustrating, the risks from leachables in inhalation products is not a single preset factor which can be expressed through a one-dimensional approach. Rather, it must be calculated and discovered through careful and deliberate exploration, with high-quality experimental design. In a nebulizer system, a dry powder system, and an aqueous nasal spray, there are unique attributes, and they are quite different from a Metadose inhaler filled with an organic propellant.
As was discovered 30 years ago, leachables are a knowledge-led problem, and therefore need a knowledge-led solution. The solution to nitrosamines and PAHs in Metadose inhalers was not to get better and better at analysing them, but rather to understand the source of the problem and eliminate it. Thus, the pharmaceutical industry turned to its suppliers and challenged them to create elastomers low in leachables, which did not contain sources of toxic substances, and now today's inhalers do not have a nitrosamine or a PAH issue. The most recent issue with nitrosamines outside of Metadose inhalers and its packaging system has perhaps its root cause once again in a lack of knowledge and understanding, rather in a lack of methods of analysis. I offer this final thought. If the problem and solution is knowledge and understanding, the focus for the future should establish processes and mechanisms for gathering and retaining information which is pertinent to the risk of leachables. This would have two components: information that informs the severity of the hazard and information that establishes the probability of that hazard. The combination of these offer much more complete protection than reliance on analytical methods, which, no matter how exquisitely sensitive, cannot fully substitute for a complete science-led knowledge base.